Designer baby. This baby's genetic makeup has been artificially selected by genetic engineering combined with in vitro fertilization to ensure the presence or absence of particular genes or characteristics. The possibility of designing a baby begins with a stem cell. Stem cells have the ability to divide and transform into certain specialized cells. For example, each new cell created can remain a stem cell or develop into another cell, such as a brain cell, muscle cell, or red blood cell. Stem cells are very valuable when they do not have a determined function and are still undifferentiated. Scientists today are exploring how these undifferentiated cells become differentiated. They are trying to control how these cells develop by manipulating and inserting specific genes into them. Scientists have created many recipes for the creation of particular cell types. By studying this, important information about human development may be discovered. For instance, these cells can potentially be used to treat disease. If stem cells could be organized to differentiate into desired cell types, they could be used to replace the defective cells and tissues of many diseases, such as Alzheimer's, heart disease, and diabetes. Soon, scientists may be able to accurately direct the differentiation of embryonic stem cells into specific cell types, not only to treat diseases, but to design babies with preferential characteristics. Currently, a reformed method of designing a baby is used by many parents at risk of passing on defective genes to their children. The technology called PGD, or pre-implantation genetic diagnosis, helps people in selecting an embryo without serious genetic disorders. A collection of embryos are first created by in vitro fertilization. They are then grown to the 8-cell state, at which point several cells are removed and analyzed for the genetic variants related to the disease. Finally, the embryos lacking evidence of these harmful variants are the only ones placed in the womb. This process was first used in the 1990s and has been successful in preventing the birth of children with several serious diseases. Similar to genetic diseases, aesthetic traits are associated with numerous genetic variations. It must be decided where the line between acceptable and unacceptable should be drawn if these possibilities are exploited. Is there a moral distinction between treating or preventing disease and enhancing traits? Many people approve of genetic alteration to eliminate the chance of disease, but they do not believe in cosmetic enhancement. However, the diseases selected as suitable to be treated or prevented by genetic technologies are hard to find. While the majority of society believes these diseases to be the ones negatively looked upon, others with more extreme views believe that homosexuality and even practicing a religion different from their own to be diseases. Similar to the problem of deciding which diseases are acceptable for PGD, there is no definition of what is a better trait. Defining superior traits is very much based on personal opinion and is driven by our culture. It is going to be possible to choose traits, not to avoid disease, but because somebody has a preference for a particular child. The future genetic altering can lead to is daunting. Genetic enhancement should be rejected because of its correlation with the former eugenics programs endorsed by the Nazis. Adolf Hitler wanted to eliminate the weak in the German population. The Nazis used the science of eugenics to substantiate their prejudices intended to solve the nation's troubles by eliminating the inferior racial traits of society. The Nazis' extreme distortion of eugenics was an important factor leading to the horrific Holocaust. If we continue to let individuals design babies, there will be a repeat of history. Preferences for babies with certain traits will create a society in which those without the traits will be deemed inferior. Scientists are once again manipulating the study of genes into an opportunity to perfect our offspring.